Fly Lab, final presentation, Exploring Mutations in Drosophila melanogaster by Elias Uketsu and Suyash Goswami.
This slide introduces the two unknown eye color mutants we studied in Drosophila melanogaster, which are pig and pit. Figure 1 displays the three eye phenotypes side by side to help us visually distinguish them. Image A shows the wild type flies with the darker red eyes, which serve as the baseline comparison. Image B features the pig mutant, which has bright red eyes that are similar to wild type, just slightly lighter. Then image C shows the pit mutant, which have yellow eyes and are distinctly different from the other two. These visual differences were crucial for tracking phenotypes across generations and determining if each mutation was likely X-linked or autosomal, which we will further explore in the results and mapping slides to follow.
For this experiment, we isolated 9 virgin females from both mutant colonies. We went on to crossing these mutant females with wild-type males. We did this as it allowed us to conclude whether our mutations were X-linked or autosomal. If the mutation were autosomal, then all of the offspring would express the wild-type phenotype. This is because we would be crossing a homozygous dominant, the wild type, with a homozygous recessive, the mutant. The offspring would all be heterozygous and therefore would all express the wild type phenotype. However, if the mutation were to be X-linked, the male offspring would express the mutant phenotype, while the female would express the wild type phenotype. This is because the males are getting they are getting their only X chromosome from the mutant mother, which would automatically make them mutants. On the other hand, the females would be getting one mutant X chromosome from the mother and the other wild type X chromosome from the father, making them heterozygous. To isolate these flies, we first had to clear the vials. After waiting 6 hours, which allows the larvae to hatch and for the females to still be virgins, we proceeded to etherize them. And then we went on separating them into males and females based on whether or not they had sex combs. And after finding all of the females, we put them in with the wild type males in a new flask and removed them after 7 days to let them breed. The results allowed us to see whether they were going to be X-linked or autosomal.
This slide shows the F1 generation results from two separate crosses between wild-type males and virgin mutant females. Table 1 refers to pig and Table 2 refers to pit. As shown in Table 1, all 19 offspring that were collected all had the wild-type dark red eye phenotype. None showed the mutant bright red eye phenotype. And in the mutant cross in Table 2, all 21 offspring had the mutant yellow-eyed phenotype and none showed the wild-type phenotype. As stated before, if it were X-linked, the mutant phenotype would only be expressed in the males, not female flies, and if it were autosomal, all offspring would express the wild-type phenotype. Since all flies for the pit mutant expressed the same mutation, there was likely contamination of the vial, making the determination of X-linked or autosomal not possible for this cross.
We chose our pig mutant for a 3-point mapping. This mutant had bright red eyes and an autosomal mutation. We knew this because all the offspring expressed the wild type phenotype. We would have done our 3-point mapping with a test cross by using the F1 generation, the heterozygotes, and more homozygous recessive mutants. This will allow us to determine the location of each gene relative to each other and the distance between them. This is our simulated data for a 3-point mapping. The double crossover was between U, L, pin wild type, U wild type, L wild type, and pin, as those had the lowest frequencies, and the parent chromosomes were U wild type, L, pin, U, L wild type, pin wild type, as they had the highest frequencies.
This slide shows the genetic map of the two other genes along with the pig mutant. After calculating the distance between each gene using our simulated 3-point mapping results, we found that the middle gene was L. The pig mutation was shown to be 18 map units away from L and 55 map units away from pig. This slide represents our working hypothesis regarding the pig mutation. Based on the observed phenotype, which is bright red eyes, we hypothesized that the pig mutation may be on a gene that codes for cinnabar eyes, also known as CN, located on chromosome 2. The rationale for this hypothesis is that the cinnabar phenotype is also characterized by bright red eyes, which closely resemble the pig mutant. Secondly, according to the chromosome map, the CN gene is located at 57.5 map units, which aligns closely with our simulated mapping data for pig, which showed a total of 55 map units.
Our complementation cross would have been between the pig mutants and the cinnabar eye mutants. If the mutations had been on the same gene, then the offspring would have been homozygous recessive and would have expressed the mutation. However, if the mutation was on different genes, then the offspring would have been heterozygous and would have expressed the wild type phenotype. If our hypothesis is correct, then the mutations would land on the same gene, causing all offspring to be mutants. If it's incorrect, then the offspring would have the wild type phenotype.
Based on the data, we have concluded that the pig mutation is autosomal. Since all of the offspring were wild type, including the males, the mutation could not have been X-linked. However, our data for PIT was inconclusive since our results did not fit X-linked or autosomal. And from the data collected, we can hypothesize whether the pig mutation is on the CN gene.
The cinnabar eyes gene codes for the enzyme 3-hydroxykynurenine, which is important for the synthesis of omochromes, also known as brown color pigments. What we would want to know is how different types of mutations, such as frame shifts or point mutations, would affect the gene differently. Would one cause a weak loss of function while the other cause a strong loss of function? Would both just result in null mutation? To test this, we want to use an intercalating agent and an alkylating agent on the cinnabar eyes gene. First, we would want to use fish, and since we want to test the effects of these agents on our specific genes, we would want to open up the DNA using fish so we could see into it. Firstly, we would grow two identical cultures of flies. Then we would find and open up that part of the DNA with a CN gene, the cinnabar eyes gene, and we would see what the effects of the intercalating and alkylating agents are on the phenotype, and we could then differentiate them.
Contributions. So both Elias and CR split the work up equally, and both partners sometimes together and individually isolated and crossed the version females with the wild type males. Both partners met up in person to analyze data and finish the slideshow, also reviewed and corrected each other's work while making the FlyLab slides. And these are our references.